Mitosis. So I'm going to talk about mitosis first, and then I'm going to go over meiosis. So the main process of mitosis and why cells go through mitosis is that they need to create new cells. So it could be because of dead cells, it could be to replace damaged cells. But if you recall from cell theory, all new cells come from existing cells. So in order for new cells to be created, they need to go through mitosis, which is cell division. So, mitosis specifically occurs in all your body cells. So, that's all cells in your body, all cells in living organisms, except for the sexually producing cells. So, testes and ovaries go through a separate process in order to create your sex cells. So, in mitosis, we start out with one cell, and the end product at the end of mitosis are two cells. So, both of these cells created at the end of mitosis are diploid. So, the term diploid means that they contain two sets of chromosomes. So, they have two sets of chromosomes, one from mom, one from dad. So, diploid cells have two sets of chromosomes. And then also, these cells created at the end of mitosis are genetically identical to the original starting cell, which means the DNA is identical. So if you think about all your skin cells in your body, they are all genetically identical to the original skin cell that it started from before mitosis occurred. All right, so before mitosis occurs, we have what we call interphase. So this happens before mitosis. So if you look at the yellow here, that's interphase. And then we have mitosis, which is in green. And then we have cytokinesis, which happens after mitosis. So in the cell cycle, we have interphase happening first, and then mitosis, and then lastly, cytokinesis. So if you take a look at interphase, in G1, this is when your cell is growing in size. In synthesis or S phase, this is where your DNA is created and the chromosomes are replicated. So if you take a look at this picture on the right, here you have the original chromosomes inside the starting cell. During interphase, they are replicated. So you make a copy of this. So now we have two identical strands that make up a chromosome. So these two identical strands that contain identical DNA, because you essentially made a copy, are called sister chromatids. So these two strands are sister chromatids. And then last step of interphase is G2. And during this step, this is where the organelles necessary for cell division are produced. All right, so you can summarize mitosis into the acronym PMAT. So PMAT stands for PMAT. The first one is prophase. So the first stage of mitosis is called prophase. And in this stage, we see that the nuclear envelope starts to fall apart. Your chromosomes start to move around, and then you have spindle fibers that start to form. So, spindle fibers are necessary in making sure that the chromosomes separate correctly. Metaphase, M metaphase. Chromosomes are lined up at the middle of the cell. So spindle fibers are helping the chromosomes line up at the middle of the cell. Anaphase happens after metaphase, and this is when your sister chromatids, these are identical, separate and move apart from each other. So they move to opposite ends of the cell. The centromeres and centrioles and spindle fibers that were used for mitosis start to disappear in anaphase. In telophase, we see that there are two nuclear envelopes that start to form around the chromosomes. So in telophase, this is right before the cell divides in half. So we see that there are two nuclei, two nucleus, one on each side of the cell. So when we divide the cell in half, each of those cells will have their own nucleus. So cytokinesis is when the cytoplasm actually divides. So from here, we are in one cell with two nuclei. And then cytokinesis is when the cytoplasm, the empty space inside the cell, actually divides. So in an animal cell, we call that a cleavage furrow. And then in a plant cell, we call that a cell plate. So keep in mind the two cells created at the end of mitosis have the same exact DNA as the original cell before it split. All right. So a quick way to memorize mitosis, PMAT, PMAT. interphase happens before mitosis, and then cytokinesis happens after mitosis. Moving on to meiosis. So, meiosis is slightly different. So, first of all, it doesn't occur in all body cells, it only occurs in your sex producing cells. So, testes and ovaries. Testes and ovaries go through meiosis to produce your sex cells, which are egg and sperm. So, in meiosis, there's a typo here. In meiosis, we end up creating a total of 4 cells at the end of cell division. So in meiosis, we create a total of 4 cells. Each of those cells are haploid, meaning that they only contain one set of chromosomes. And then these cells are not genetically identical, but genetically different, which means the DNA is different from the original starting cell. So all 4 cells created at the end of meiosis are genetically different, contain different DNA than the original starting cell. So that just tells you that all the sperm and all the eggs in your body are all genetically different, which makes sense because when you have a kid, you're not going to have clones of you, or you're not going to have clones of the kid itself. All right, so meiosis occurs in our ovaries and testes. Ovaries produce eggs and testes produce sperm. And once again, we end up creating 4 genetically different cells. And these cells are haploid, so they only contain half the number of chromosomes as the original. So, for example, normal diploid cell in a human body has two, two pairs of 23 chromosomes, so 2 sets of 23 chromosomes. A egg or sperm cell would only have 23 chromosomes, so they would only have 1 set of 23 chromosomes. All right, so one big thing in prophase 1 of meiosis 1 is that we have crossing over occurs. So homologous chromosomes, one chromosome comes from the mom, one chromosome comes from the dad. They contain the same set of genes. They're going to cross over. So segments of the chromosome cross over each other. When that happens, these segments of DNA are exchanged. So now you have parts of the mom DNA and parts of the dad's DNA on these new chromosomes. So homologous chromosomes crossing over only occurs in prophase 1 of meiosis 1. Metaphase 1. So anytime you see metaphase, you always see them lining up in the middle. So here we have the homologous chromosomes lined up as a pair in the middle of the cell. So think of it as 2 lines instead of a single file line. So they line up next to their pair. These are called homologous chromosomes. They are a pair. So they're going to line up at the middle. In anaphase 1, we see that the homologous chromosomes get pulled apart to the opposite ends of the cell. And then again, in telophase 1, we can see that the nuclear envelopes or nuclear membranes start to form. And we see that there are 2 nuclei in a single cell. So in telophase 1, we're still connected in a single cell. In cytokinesis is when the cytoplasm actually divides in half. All right. And then meiosis 1 carries on to meiosis 2. So meiosis is actually 2 rounds of cell division. So these 2 new cells are going to go into meiosis 2. So in meiosis 2, prophase 2, we can start seeing the sister chromatids. So the 2 strands start to appear on the chromosome. So before it was homologous chromosomes, but after they were split apart, they no longer have their homologous pair. So now we see that the 2 sister chromatids start to appear in the chromosomes. In metaphase 2, the chromosomes are lined up at the middle. So this time it is a single file line because there's no more homologous chromosome pair. In anaphase 2, we see the sister chromatids now being pulled apart. So instead of the homologous pair being separated in anaphase 1, in anaphase 2, your sister chromatids begin to separate to opposite ends of the cell. And then telophase 2, we end up forming our 2 new nuclei again. So each of those end up creating 2 new nuclei. So we end up with 4 haploid cells. So the reason why it's called haploid is we only have 1 set of chromosomes. So before in the starting cell, there were 2 sets of chromosomes, meaning that there were 2 of the short one and also 2 of the long one. you can see in the final cells created in meiosis, there's only one set. So the cells created at the end of meiosis are haploid. And if you look at all the coloring here, they have a little bit of mom, a little bit of dad in each of the cells, but none of them look the same. So this is what we mean when we say genetically different. So genetically different cells. So once again, eggs and sperm are created through this process.